Good morning. Today our topic is photosynthesis. In photosynthesis, light energy is converted into chemical energy and it is stored in the form of organic compounds. And photosynthesis, this process occurs in plants. They are autotrophic, they can synthesize their own food. And in the plants, leaf is the main organ in which there is chloroplast. And in the chloroplast, we know there are thylakoids, and in this organelle, light energy is absorbed. And in the light energy, we know the reasons, that is visible reason. It is mainly absorbed, and in the visible spectrum also there are only specific wavelength of light which are absorbed by these pigments. And when light is absorbed, organic compounds. Next, photosynthesis occurs within the stacks of flat disc called grana. These are granas. It is located in the stroma of plant cell organelles. It is the structure of chloroplast. This is the chloroplast structure. This is the outer membrane. This is the inner membrane. This extracellular matrix, and this is the stroma. And these are the thylakoids, and it is the stacks of grana. And in this, light energy is mainly absorbed. And there are two stages in the photosynthesis. One is the light reaction, second is the dark reaction. Light reaction, absorption of light energy. And the second is the dark reaction. Dark reaction involves synthesis of sugar molecules, and it occurs mainly in the stroma. It is not directly dependent upon light, but it is indirectly dependent upon light. And in the photosynthesis, 6 molecule of carbon dioxide plus 6 molecule of water in the presence of light, and light is absorbed by chlorophyll pigment, and there is formation of glucose and release of oxygen molecule. Next, photosynthetic pigments, they are located in the thylakoid membrane of chloroplast. They absorb light energy and convert it into chemical energy. Chemical energy in the form of adenosine triphosphate. They absorb only specific wavelength of visible light. For example, only the red and this bluish light, it is absorbed by chlorophyll molecules. By reflecting others, in the case of this chlorophyll molecule, green light is mainly reflected back. That's why the leaves are of green color. There are majorly three types of photosynthetic pigments, namely chlorophyll, it is a fat soluble pigment; carotenoid, it is also fat soluble pigment; and phycobilin. Phycobilin mainly occurs in algae. These are types of different pigments. Chlorophyll is further categorized into chlorophyll A molecule, chlorophyll B molecule, chlorophyll C, chlorophyll D, and these molecules are present in bacteria: bacteriochlorophyll A, bacteriochlorophyll B, and chlorobium chlorophyll A and chlorobium chlorophyll B. Chlorophyll A and B, it is majorly present in all photosynthetic plants, and it is present in plants as well in lower plants, that is algae, but it is not present in bacteria. In bacteria there is no presence of chlorophyll A molecule. Chlorophyll B molecule is present in plants and green algae. Chlorophyll C is present in brown algae and diatoms. Chlorophyll D is present in red algae. And bacteriochlorophyll A and B, that is present in bacteria, that is purple and green bacteria and purple and sulfur bacteria. Chlorobium chlorophyll is present in green and sulfur bacteria. The second group of photosynthetic pigment is carotenoids. Carotenoid is further divided into two categories, that is carotenes and xanthophyll. Carotene, it is present in algae and higher plants. Xanthophyll, it is also present in algae and higher plants. Phycovalence is further divided into phycoearthine, phycocyanovulin. It is present in blue-green algae and red-green algae. Now we will discuss about this chlorophyll molecule. It is the green pigment occurs in photosynthetic organisms that is responsible for absorption of light energy, and this light energy is converted into chemical energy for photosynthesis process. Chlorophyll is the primary light absorbing pigment, and it mainly absorbs violet, blue and red waves of visible region of sunlight, and it reflects back the green color. Chlorophyll is a fat soluble compound essentially made up of two components, that is porphyrin ring and phytol. Porphyrin rings have 4 rings and phytol is long carbon chain structure. Chlorophyll molecules are located special cellular bodies, that is chloroplast, and it occurs as bonded to the proteins. Chlorophyll is formed from protochlorophyll in the light, and addition of 2 hydrogen atoms to protochlorophyll gives rise to chlorophyll molecules. These are leaf, and this is the section, and in the section we can see here are chloroplast molecules, and this is the incident light energy from the sunlight, and visible spectrum bigger, and we know the reflected light is greenish color, and this is the absorption of light energy. It gives rise to absorption spectrum. This is the chlorophyll B molecule, and this is the chlorophyll A molecule absorption spectrum, and this is the wavelength, chlorophyll A molecule wavelength, and this is the chlorophyll B molecule wavelength. Chlorophyll structure is made up of 4 pyrrole, that is tetrapyrrole. 4 means tetrapyrrole, which is also known as porphyrin. We can see 1, 2, 3 and 4, and this is the phytol. This one is the phytol tail, and there is a difference between the chlorophyll A and B molecule only in the position of this group. In the case of chlorophyll A molecule, it is methyl group, and in the case of chlorophyll B molecule, it is the aldehyde group. It is in the chlorophyll B. It is in the case of chlorophyll A molecule. The remaining structure is the same. The magnesium is present in the center. It is present in the center and bonded with this nitrogen, and this is the covalent bond. And chlorophyll A molecular formula is C55H72O5 and 4 magnesium, and in chlorophyll B molecule is C55H70O6 and 4 magnesium. It is blue green in color, it is yellow green in color. It is a primary pigment, and it is an accessory pigment. Sorry, it has a methyl group attached to the 3rd carbon, and it has aldehyde. It is soluble in petroleum ether, and it is soluble in methyl alcohol. The second group of photosynthetic pigment is carotenoids, and they are the secondary light absorbing pigments. It is also known as accessory pigments. They are present in the thylakoid membrane, and they are supplementary light receptors as absorb blue light and play role in dissipating of excessive radiant energy. They reflect red, orange and yellow light. We know in the case of chlorophyll molecules, reflection of green light, but in the case of carotenoids, reflection of red, orange and yellow. That's why a number of means plant products like tomato, pumpkin, they have a red, orange, yellow colors, and also some capsicums. They are very long chain water and water repelling. It means it is also fat soluble. And there are two types of carotenoids, that is carotenes and xanthophyll. Carotenes are orange color, xanthophyll are yellow in color. They protect the chlorophyll against photooxidation and then transfer the light energy to the chlorophyll. It also helps in preventing photodynamic damage. They are able to express themselves in mature leaf even if the chlorophyll begins to disintegrate. These are groups: carotenoids, carotene and xanthophyll. Carotene, example beta carotene, alpha carotene, and xanthophyll, leucine, zeaxanthin, capsanthin, astaxanthin. These are all example of carotenoids, and these are the leaves. It contains carotenoids, that's why it is of yellow color. And sunflower, it also contains the pigment carotenoids, and these all things are capsicum, corns, apple, tomato. These all things also contain carotenoids pigments. And the difference between the carotene and xanthophyll is also in structure. Alpha carotene and beta carotene, it doesn't contain any oxygen group, but in case of xanthophylls like zeaxanthin, astaxanthin, leucine, they contain oxygen group. We can see this oxygen. Next, the next group is phycovalents. These are open chain tetrapod structure. We can see here, this is open chain. There is no close, means there is no magnesium ion present here, and it is present in green algae, red algae, and there is phycoerythrobilin and phycocyanubilin. They are mostly present and they harvest light. Phycovalents are covalently linked to specific binding proteins, forming phycobilin proteins which associate to form highly ordered complexes called phycobilisomes. They are the primary light harvesting structure in the microorganisms like green algae and red algae. Next. Pigments absorb light as a source of energy for photosynthesis. The absorption spectrum indicates the wavelength of light that is absorbed by a particular pigment. Means particular wavelength is absorbed by this pigment, and this is the absorption spectrum. The action spectrum indicates the overall rate of photosynthesis at each wavelength of light. We can see here, this is the visible light region of sunlight, okay, and light is absorbed by pigment in the chloroplast molecule. This is the thylakoid, and this is grana, and this is the stroma. Light is absorbed here, and green light is reflected back, okay. This is the absorbance, this is the photosynthetic rate. Photosynthetic rate action means what happens after this absorption. You know, this one graph, and absorption at this wavelength, particular wavelength. This is the wavelength, and in the case of carotenoids, this is for carotenoids, chlorophyll A molecule, chlorophyll B molecule, and this one is the action spectrum. An action spectrum shows the overall rate of photosynthesis at each wavelength. Okay, both display two main peaks, a large peak at blue region 450 nanometer we can see, and another smaller peak at the red region 670. This is the here, we know red region is around this, and one is the bluish region. here. Okay. Both displayed off in the green and yellow portion of the visible spectrum. You know, this is the green. There is a drop of this molecule. This is again same differences. Graphic representation of degree and portion of light wavelength absorbed by a substance is called absorption spectrum, and action spectrum depicts the effectiveness of light absorbed of various wavelength in performing a particular function for that is photosynthetic function. And a recording of a wavelength of light absorbed by a substance is made up with the help of an instrument that is spectrophotometer. And in absorption spectrum, the height of curve at a given wavelength gives the relative absorbance at that particular wavelength. Absorption spectra of chlorophyll A and B show that the two pigment absorbs maximum in blue, violet and red parts of light. It has been found that chlorophyll A has several forms which shows maximum absorption in red region at different wavelength like 660, 670, 676, 690 to 700 nanometer extra. Photosystem, pigment system 1 and pigment system 2. There are two pigment system, photosystem 1, photosystem 2. It is mainly present in green plants and cyanobacteria. In the case of bacteria, there is only one type of photosystem, and on ultracentrifugation, photosystem 1 is lighter fraction and photosystem 2 is the heavier fraction. We know a photosystem now present in chloroplast, and in chloroplast there is thylakoid membrane. In the thylakoid membrane, photosystems are present. This is the photosystem 2 and this is the photosystem 1. It absorbs light. Okay. And in photosystem 2, there are two photosystems present, that is photosystem 1 and photosystem 2. And on ultracentrifugation, we get the lighter fraction, that is PS1, and the heavy fraction, that is PS2. It is the heavy and PS1 is the lighter fraction. Green plants and cyanobacteria have two photosystems, that is photosystem 1 and 2, and bacteria have only one type of photosystem. It is situated in thylakoid membrane, as we have already discussed. In the thylakoid membrane, this is the photosystem 2 and this is the photosystem 1. There is a variation in chlorophyll A molecule, chlorophyll B molecules and cationates in these systems. Okay, and the photosystem 2, they have reaction center different, and photosystem 1 have different reaction center. Photosystem 1 have reaction center that is 700 and it has 680, and both of these photosystem absorb light. In the PS2, there is photosynthesis of water, breakdown of water molecule, and there is release of oxygen molecule, and there is electron molecule which are transferred from different types of plastocanin, cytochromes and plastocanin molecule, and it is further electrons are transferred to photosystem 1. There are two type of electrons in the case of this photosystem 1. One is coming from this light and another is from photosystem 2, and they are further transferred to ferrotoxin and NADP reductase, and there is reduction molecule, and it is in the inside the stroma site. Okay, and they say there is transfer of electron molecule, and in this thylakoid space there is also a ATP synthase molecule, and in this ATP synthase there is phosphorylation reaction and production of adenosine triphosphate by pumping out through these ions. And next, This is the difference between photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2 both are situated in thylakoid membrane. Photosystem 1 have 200 to 400 molecules of chlorophyll molecule and 50 catenoids. We know already discussed this 50 catenoid molecule, and reaction sector is P700, that is chlorophyll A, and plastocyanin, cytochrome F, 2 molecules of cytochrome B6 and 2 molecules of ferredoxin. This is present in photosystem 1, and photosystem 2 is constituted of 200 chlorophyll molecules, 50 carotenoids and 1 molecule reaction center, that is P680, and there are substances like zinc, magnesium, calcium, cytochrome F, plastocyanin, etc. The number of chlorophyll B molecule is more in photosystem 2, and in the chlorophyll A molecule, photosystem 1 chlorophyll B molecule number are literally less in comparison to photosystem 2. Reaction center is P700, here reaction center is P680. It is located on the outer surface of thylakoid. In the outer surface of thylakoid. We know this is the on this outer surface, and located, this photosystem 2 is located on the inner surface of thylakoid membrane. This is the inner surface. Here it is photosystem 2. In PS1 molecule, oxygen 2 is not evolved. In photosystem 2, molecular oxygen is evolved. Pigment system 1 formed NADPH, and NADPH is not formed in the photosystem 2. Photosystem 1 participate in both cyclic and non-cyclic photophosphorylation. There are two types of photophosphorylation, cyclic and non-cyclic. Both are present in photosystem 1, but photosystem 2 has only one kind of photophosphorylation, that is non-cyclic. We can see here, this is the photosystem. This is a chloroplast molecule. In chloroplast, this is the thylakoid, and this is the chloroplast envelope, and it is the grana molecule, and this is the stroma, stromal thylakoid, and this is the stroma, nucleate, plastone, ribosomes, starch granules. Okay, starch is synthesized here, and we have two photosystems now, photosystem 2 and photosystem 1. PS1 is reaction center is P700. There is less number of chlorophyll B molecule in comparison to photosystem 2. Chlorophyll A molecules are more in this system, photosystem 1. There is exchange of energy in both photosystem 1 and 2 during this photophosphorylation reaction. We can see here also there was one point that photosystem 2 is situated in this side, photosystem is situated on another side. This is the thylakoid. We can see this portion is the inner portion of thylakoid. Here, look here. You can see here. Photosystem 2 is mainly in inner side, and photosystem 1 on the outer side. See, this is the outer side of thylakoid. Here present photosystem 1, and photosystem 2 is mainly present in inner side, and also in this stromal thylakoid. Here also you can see this is not inner side, this is the outer side. So the here also present photosystem 1, PS1. Okay, next. There are two types of photophosphorylation. Photosynthesis in the presence of light, and We know that there are two types, one is cyclic and one is non-cyclic. Cyclic photophosphorylation, in cyclic photophosphorylation there is only photosystem one is involved, and electrons released by photosystem one is recycled by this photosystem. Only ATP is produced and no photolysis of water is take place here, and no oxygen is released in the cyclic photophosphorylation. In non-cyclic photosystem 1 and 2 both are involved. Electrons are not recycled here. Both ATP and NADPH are produced. Photolysis of water also takes place here. Oxygen is released due to photolysis of water. We know this is water, and from this oxygen is evolved. Electrons and protons also. And we know this is the cyclic photophosphorylation. In the cyclic photophosphorylation, only photosystem 1, PS1. So reaction center will be P700. This is the photosystem 1, and there is when light is absorbed, chlorophyll molecule get excited and electrons are transferred, and from here electrons are transferred in this redox chain reaction. There is two steps: oxidation and reduction. Oxidation reduction step, ferredoxin, plastocaine, cytone, plastocyanin, and then further electron. It is then again to the chlorophyll molecule. Here comes chlorophyll molecule, and there is production of only ATP. There is no production of any other SNBT NADPH. And photosystem 2, it is there is Z scheme, non-cyclic photophosphorylation. The photosystem 2 and photosystem 1 both are involved in this, and redox reaction also taking place in reduction, oxidation. Photon molecules, this is the light energy, and chlorophyll electrons are transferred, excited state, and electrons are further transferred in this, and plastocaine, cytoplastocyanin, and there is production of ATP molecule. There is photolysis of water also, that is water molecules break down and oxygen is produced here, and electrons are further transferred into photosystem 1, and photosystem 1 involved in ferredoxin, and there is production of this NADPH molecule. Next, photophosphorylation, it is a synthesis of ATP in the chloroplast, and it is a light driven process of electrons from the water to the NADP coupled to the translocation of proton from the stroma across the thylakoid membrane. This is the, we know, this is the stroma region, and this is the thylakoid, this is the chloroplast. This is the chloroplast molecule. Okay, and in chloroplast it is the stroma region, and this is the thylakoid, and this is the thylakoid lumen, and in this thylakoid membrane there is presence of photosystem 1 and photosystem 2. Okay, active site of oxygen involving enzyme is arranged so that proton formed during water oxidation are released into the thylakoid lumens. We know there is photolysis of water, and it is inside the thylakoid lumen. So these protons molecule contribute to the electrochemical proton potential gradient. The electrochemical potential gradient is formed, and the thylakoid membrane contains a protein that function to transport chlorine across the membrane. Proton accumulation in the thylakoid lumen is electrically balanced with the part of by chlorine uptake. There is uptake of another molecules, and gradient is maintained. Thylakoid accumulated HCl, and the membrane potential across the membrane is low. The pH inside the lumen during steady state photosynthesis is about 5.0. Here in the thylakoid interior, this is the lumen side. There here pH is low and high concentration of proton, and in the stroma side there is there is high pH and low concentration of proton ions, and there is ATP synthase molecule here. ATP synthesized in the stroma side, and this ATP is further utilized in the further formation of organic compounds. And now we will study dark phase. In the dark reaction, we will study three things, that is C3 cycle, C4 cycle, CAM cycle, and in all these, all of the synthesis of organic compounds take place. First is the Calvin cycle. Calvin cycle is also known as C3 cycle, and it is also known as reductive pentose phosphate cycle. Because pentose sugar is involved here, it is a light independent process. It is not dependent upon light directly, but indirectly it depends upon light, because ATP formed in light reaction, it is utilized here, and it takes place in the stroma of chloroplast. We know that light reaction takes place in the thylakoid, and the synthetic phase occurs in the stroma region of chloroplast. Carbon dioxide is fixed here. In the case of this dark reaction, carbon dioxide is fixed by a series of chemical reactions to form carbohydrate. The energy for these reactions come from ATP and NADPH, which are formed during photophosphorylation reactions, that is cyclic and non-cyclic photophosphorylation reaction, and it takes place in the three key steps. C3 cycle mainly have three steps, that is carbon dioxide fixation, reduction of carbon, and regeneration of IUPAP. It is 5 carbon compound. That's why it is also known as pentose phosphate cycle, and reduction takes place, and this is a carbon dioxide molecule, 3 molecules of carbon dioxide, and there are 3 molecules of ribulose 1,5 phosphate, 1, 2, 3, 4, 5. There are 5 carbon molecule compound, that is ribulose 1,5 biphosphate. On the 1 position there is phosphate, and in the 5th position there is phosphate. And this is a RUBP molecule, and this RUBP molecule with this react with this 3 molecules of carbon dioxide in the presence of enzyme viscose, and this enzyme help in carbon fixation, and there are generation of 6 molecule of 3 phosphoglycerate molecule, 6 molecules of 3 phosphoglycerate. It is further reaction, and there is production of 6 molecule of 1,3 biphosphoglycerate molecule, and there is further reduction, power is utilized, NADPH is used to NADP, and there is 6 molecules of glyceride 3 phosphate molecule, and this glyceride phosphate molecule, 1 molecule, 6 molecule of glyceride 3 phosphate is formed here, 1 molecule is utilized for the sucrose and other compound, and the 5 molecule of glyceride 3 phosphate, this is utilized for the regeneration of ribulose 1,5 phosphate, and there is utilization of ATP molecule and formation of ribulose 1,5 biphosphate. So this Calvin cycle mainly in 3 steps. One is carbon fixation, second is chemical reshuffling through this 3 biphosphoglycerate to glyceride 3 phosphate molecule, and further is regeneration of a IUP molecule by 5 molecules of glyceride 3 phosphate. And the glucose synthesis reaction is 6 carbon dioxide, 6 IUP molecule, 12 NADPH, 12 ATP, 10 H2O, and this gives rise to 4 GAP, 2 DHAP, fructose 6, and this is the reaction. And regeneration of IUP, that is by this, and net reaction from 6 turns of Calvin cycle is 6 carbon dioxide, 12 NADPH, 8 molecules of 18 molecules of ATP, and 12 molecules of water. It gives rise to glucose molecule, 12 NADP and 18 ADP, and 18 inorganic phosphate molecules. This is the net reaction. We can see here, this is the carbon dioxide molecule, and this is the Rubisco enzyme. This is the 5 carbon compound ribulose 1,5 biphosphate. It will give rise to this 3 phosphoglycerate, then dehydrogenase activity, and glyceride 3 phosphate molecule are utilized here. Look here, glyceride 3 phosphate, it is further transcriptolase, and Further, there is glycerol phosphate. It is dihydroxyetone DH. It forms DHAP, dihydroxyacetone phosphate. One glycerol phosphate. It will form erythrose 4 phosphate, and there is one dihydroxyacetone phosphate, and this dihydroxyacetone phosphate, it is exported and it is aldolysis. This is glycerol phosphate. It is converted into dihydroxyetone phosphate. It will form the aldolase molecule, this fructose 1,6 base biphosphate with the help of enzyme aldolase, and again there is phosphatase enzyme involved, and it forms fructose 6 phosphate, glucose 6 phosphate, glucose 1 phosphate, and further it leads to formation of a starch molecule. This is the stromal hexol phosphate pool, and further this fructose 6 phosphate, it can also utilize to form trans-ketolase reaction. And xylose 5 phosphate molecule, 5 carbon compound is formed, and erythrose 4 phosphate compound is also formed. It reacts with this dihydroxyetone phosphate with the help of enzyme aldolase, and it forms pseudoheptulose 1,7 biphosphate. 4 carbon this, 3 carbon this, 4 3, and pseudo is 7 carbon, 4 plus 3, that is equal to 7, and there is ketonic heptulose, and there is phosphatase. It is 1,7 biphosphate, and 1 phosphate is removed by phosphatase enzyme, and it will give rise to pseudoheptulose 7 phosphate. This pseudoheptulose 7 phosphate and glycerine 3 phosphate, it is further react with the help of enzyme trans-ketolase and form 5 carbon compound, that is ribose 5 phosphate, and 5 carbon compound, that is xylulose 5 phosphate. It is catalyzed by trans-ketolase, and xylulose is further with the help of epimerase enzyme ribose 5 phosphate. Ribose 5 phosphate is isomerase with the ribose 5 phosphate, and there is further kinase enzyme activity, and ribose 1,5 phosphate molecule is regenerated here. Next, C3 plants occur in C3 cycle occurs in C3 plants, and C4 cycle, it is also known as Hatch and Slake pathway dicarboxylic acid cycle, and it is present in C4 plants. C4 plants mainly occurs in dry tropical region, and examples are sugarcane, maize, and grass. In C4 plants, there are specific type of anatomy, that is Kranz anatomy, and it was characterized by Korsakh 1964. He gave this name Kranz anatomy. It means a ring or neck or necklace. In this Kranz anatomy, vascular bundle is surrounded by thick walled bundle sheath cell, and it is called Kranz. Cells have large number of dimorphic chloroplast. There are 2 types of chloroplast, and it varies on the basis of structure and size, and it is thick walled. The chloroplast of bundle sheath cell have less number of grana as compared to mesophyll cell chloroplast. Dimorphic chloroplast prevent the photorespiration. Photorespiration is a mechanism where there is utilization of oxygen and release of carbon dioxide in the presence of light. In the C4 plant, there is dimorphic type of chloroplast, so it is prevented, and it increases the tolerance of high temperature in these plants. First product of 4 carbon compound, that is oxaloacetic acid. It has 2 carboxylic group, that's why it is also known as dicarboxylic acid cycle. We can see here, this is the upper epidermis, this is the lower epidermis, and this is the stoma. Through stoma there is exchange of gases. From this leaf structure part, stoma, there is exchange of gases. We can see here, this is the vascular bundle, and this cell, it is a ring like structure, that is bundle sheath cell with special type of chloroplast. This is bundle sheath cell, okay, and this is chloroplast, and this is xylem and phloem, And these are mesophyll cells. Mesophyll cells also have chloroplast. Bundle sheath cell also have chloroplast. So there are two types of chloroplast. We can see here. This is the atmospheric carbon dioxide. It enters through stroma, and this is the mesophyll cell. This cell, no, first cell. These are mesophyll cell, and these are bundle sheath cell. These are cell. These and these mesophyll cells, bundle sheath cell. Atmospheric carbon dioxide, and there is phosphenol. This is the plasma membrane cell wall. Phosphenol pyrite, and this is the bicarbonate fixation. And C4 acid means 4-carbon oxaloacetic acid is formed in these mesophyll cells, and it is transported through plasmodesmata into the bundle sheath cell. Here it is further decarboxylated, carbon dioxide is released, and this carbon dioxide is in this bundle sheath cell is utilized in C3 cycle. Here occurs C3 cycle. So C3 cycle occurs where? In C4 plants also there is C3 cycle, but it does not occur in the mesophyll cell. It occurs in the bundle sheath cell. Okay, and this acid is further transported, and there is regeneration of C2 acid. There is again this cycle performed. Next. Here we can see here. This is the atmospheric carbon dioxide and phosphenol pyrite carboxylate, and the formation of 4-carbon acid, that is oxaloacetate. Oxaloacetate is further converted into 4-carbon acid, that is malic acid. Malic acid is again malic acid, again through malic enzyme there is 3-carbon compound is formed, that is pyrite, and there is release of 1 carbon in the form of carbon dioxide, and this carbon dioxide is utilized in this Calvin cycle. And the first step in the mesophyll cell, where phosphenol pyrite carboxylase enzyme catalyze the formation of oxaloacetate from phosphenol pyrite and carbon dioxide. Okay, this is the oxaloacetate, this is the phosphenol pyrite molecule, and here it is a carbon dioxide coming from the atmosphere. Both of these are react in the presence of better enzyme PEP carboxylase, phosphenol pyrite carboxylase. Oxaloacetate reduced to malic acid in the presence of malodehydrogenase enzyme, and malic acid then transferred to the bundle sheath cell through plasmodesmata. In bundle sheath cell, malic acid is decarboxylated. Decarboxylated means one carbon is released, and in the presence of malic enzyme and release carbon dioxide. By the decarboxylation of malic acid, 3-carbon compound, that is pyrite, is formed. Carbon dioxide enter into Calvin cycle in the bundle sheath cells, and pyrite returns back to the mesophyll cells. In the mesophyll cell, pyrite in the presence of pyrite phosphate dikinase enzyme convert into phosphenopyrite, and the cycle repeat. The next type of dark reaction, the next type is GAN cycle, granulation acid metabolism cycle. It mainly occurs in GAN plants and in the succulents, euphorbia, pineapple, agave, cacti, moringa, orchids, aloe. All of these plants contain CAM cycle. In some hydrophytes like Hydrilla and Vallisneria, CAM cycle present here also. It was first discovered in the plants of Crassulaceae family, so it got her name Crassulaceae acid metabolism from there. They are present in dry, arid environment. CAM cycle is adapted to minimize water loss and photorespiration. Stomata are closed during the daytime and open at the night time. It inhibits transpiration and further uptake of carbon dioxide. Plant fix carbon dioxide during night and store as malic acid. Organic acid release carbon dioxide to the Calvin cycle during the daytime. You can see here stomata generally open in the daytime, but in the case of this CAM cycle, stomata are open in the night time, because in the arid region there is higher temperature. So if temperature is high, then transpiration activity will be moreover transpiration activity will in the night time stomata are open, and in the daytime stomata are closed, because it protect from transpiration. In the daytime, if stomata close, temperature is high at the outside, then transpiration will take place. So for inhibiting this transpiration process, stomata are closed during the daytime. In the night time, atmospheric carbon dioxide enters, and it enters and the phosphoryl pyrite, and it forms the this malic acid, and malic acid stored in the plant vacuoles, and in the daytime stomata are closed, and from malic acid that is stored in the vacuole, it is released by malate, and then it will form pyrite, three carbon molecule pyrite, and one carbon dioxide is releasing, and if this carbon dioxide enter into Calvin cycle, pyrite molecule will form starch in the chloroplast molecule. These are the differences between C3, C4 and chem plants. C3 plants are wheat, barley, pepper, fruit, rice and tomato. C4 are corn, sorghum, sugar and cane. Chem are pineapple, picky pear. 89% of worldwide flora are C3 plants, and C3 cycle they have. In the C4 less than 1, and scam is 10%. Typical habitat is ample distribution. C4 in warm areas, and scam is humid and tropical. First stable product in the carbon site fixation. C3 cycle has 3 carbon compound, that is phosphoglyceric acid, and malate is formed in the C4, and in the case of CAM, it is the 4 carbon compound. Anatomically, Wonder sheet cell not present, and there is without chloroplast. Wonder sheet have with chloroplast, that is Cran's anatomy. Succulents of cells and plant tissues. Photorespiration up to 40% of photosynthesis not detectable, because in the case of C4 plants there are Cran's anatomy, so photorespiration is not found here, and in the CAM plants there is a opening of stomata during the night time. Point of composition for the assimilation of carbon dioxide is 14 to 100, and this is 0 to 10, this is 0 to 10. This is the cycle here. This is the C3 cycle, C4 and CAM. In C3 cycle there is carbon dioxide entering to stomata and to the mesophyll cell. In the mesophyll cell we know there is chloroplast molecule, and there are Rubisco enzyme fix this carbon dioxide, and glyceride 3-phosphate compound is formed, and it is further in the sucrose. In the C4 plants there is phosphoryl pyrite carboxylase enzyme, and malic acid is formed in the mesophyll cells, and malic acid is further oxaloacetic acid and malic acid, and malic acid further is to the bundle sheet cell, and carbon dioxide that is released, and a phosphoryl pyrite one molecule is formed, and this carbon dioxide enter Calvin cycle, and glyceride 3-phosphate and sucrose molecule. In the CAM cycle, during the night time, stomata are open, and during the daytime stomata are closed, and here also malic acid is formed and carbon dioxide is this carbon dioxide this enter the Calvin cycle, and it form glyceride 3-phosphate and sucrose molecule. Photorespiration and dark reaction. Photorespiration take place in photosynthetic green cells. It takes place in all living cells. Photorespiration takes place only in the presence of light. Dark respiration takes place all the time. Photorespiration involves chloroplast, peroxisome and mitochondria. Dark reaction involves only mitochondria and next topic is photorespiration. It is also known as C2 or glycolate cycle. Uh, photorespiration Photorespiration involved in three organisms, that is chloroplast, peroxisome and mitochondria. Here is also the visco enzyme, but visco enzyme do oxygenate activity rather than carboxylase activity. It is a light dependent process in which oxygen uptake is concomitant with the release of carbon dioxide molecule. The inhibition of photosynthesis by oxygen is given by Otto Warburg in 1920 and also called Warburg effect. It is favored by high oxygen concentration, low carbon dioxide concentration and warm temperature. During oxygen fixation, Pg and phosphoglycolate molecules are formed, and it is catalyzed by the visco enzyme, visco oxygenase activity. There is no production of ATP and sugar molecule, although it utilizes ATP and NADPH and reduce these for the Calvin cycle. Phosphoglycolate is highly toxic and it is converted into glycolate molecule and transferred to the peroxisome molecule. It has photo inhibition. It takes place in the C3. It takes place in the C3 plants. In C4 plants, there are bundle sheath cell. In the CAM plants, their stomata is opened at the night time. It is the chloroplast. Oxygen concentration is high and diabolose 1,5-phosphate. Oxygen molecule will be fixed and phosphoglycolate molecule is formed, and this phosphoglycolate molecule phosphate group is transferred and glycolate molecule produce. This glycolate molecule converted into glycolate and then amino acid glycine, and this glycine further transferred from peroxisome to mitochondria glycine, and glycine molecule carbon dioxide and ammonia is released and serine amino acid is formed. Serine amino acid is further transferred to peroxisome and this serine converted into glycerate molecule. This glycerate molecule is utilized in this Calvin cycle. What are the limiting factors for photosynthesis? We know that light intensity, temperature and carbon dioxide concentration. These affect the photosynthesis mainly, and any of these factors that slows down the rate of photosynthesis is known as limiting factor. Like light intensity, if light intensity temperature is high but carbon concentration is low, then carbon dioxide concentration will become the limiting factor for photosynthesis. If temperature and carbon concentration is high but light intensity low, then light intensity will become limiting factor. Blackman formulated the law of limiting factor. It states that the rate of physiological process will be limited by the factor which is in shortest supply. Any change in the level of limiting factor will affect the rate of reaction. For example, this in the light reaction, rate of photosynthesis increase, but after some time it is inhibited. It will be due to any limiting factor. Carbon dioxide concentration, the rate of photosynthesis increased up to this, but again due to any limiting factor. In the case of temperature, it was high up to this, then decline. It is also due to limiting factor. Rate of photosynthesis and the intensity of light, high carbon dioxide concentration, high temperature. Then rate of photosynthesis is highest. If high carbon dioxide concentration and low temperature, it will be lower. If low carbon dioxide, low temperature, then it will be minimum. It will be higher when high concentration, higher temperature and light intensity is also increased. Next. Internal factors affecting photosynthesis are leafage, chlorophyll, hormone molecule, and accumulation of end product, that is starch, and leaf anatomy. Leafage, mature leaf, young leaves have less photosynthesis process in comparison to mature leaf, but as the leaf get sensitive, then the photosynthesis process is affected, and hormone molecules, abscisic acid suppress the photosynthesis process, but cytokines and gibberellic acid enhance photosynthesis process. as the end product is accumulated and it is not transported properly, then the photosynthesis processes also slow down. The leaf anatomy also decided the photosynthesis process. In the case of C4 plants, we know there are two types of chloroplast due to presence of bundle sheath cells and there is mesophyll cell. Then the leaf anatomy in C4 plants, it will prevent it from high heat and it will affect the photosynthesis process. In the C3 plants, there is only one kind of mesophyll cells and one kind of chloroplast, then there is more chances that the photosynthesis process is affected by other factors. And in the case of CAM plants, the stomata opens at night time only, and it will also not much more affected in comparison to C3 plants. Next. External factors include light. In the light, the type of sunlight, we know that the visible spectrum, that is mainly absorbed by pigments, and only the red and blue part of the visible spectrum, red and blue wavelength visible spectrum, that will affect the sunlight. Rate of photosynthesis is highest when both blue and red part of light are supplied together, as compared to when they are separately. Light intensity, the amount of light energy absorbed by plants, it will also depend upon the form of leaves and their phyllotaxy. Only 0.5 to 1.5% of light energy is utilized in photosynthesis and remaining is reflected back. Increase in light intensity beyond certain limit also damage the chloroplast and other cell organelles by photooxidation, and the photosynthetic apparatus are destroyed. This is called solarization. And at a certain light intensity, the amount of carbon dioxide used in photosynthesis and the amount of carbon produced in respiration will become equal. This point is known as composition point, division of light. It has been observed that plants may carry out photosynthesis when continuous light is supplied for a relatively longer period of time. Normally 10 to 12 hours duration of light availability is required to help plants to carry out sufficient photosynthesis process. Rate of photosynthesis doubles with every 10 degrees Celsius rise in temperature till the optimum value is achieved. An increase in temperature above 30 degrees Celsius results in fall in the rate of photosynthesis. The optimum temperature for the photosynthesis in C3 plants is 10 to 25 degree. In C4 plants it's higher, 30 to 45 degrees Celsius. Carbon dioxide is also one factor that affects photosynthesis. Carbon dioxide in the usually 0.03%. It is increased up to 1.0%. If the concentration of carbon dioxide is up to 1%, then the photosynthesis process will be increased from 0.03 to 1%. But more than 1%, it will be toxic for plants. And gaseous exchange stop due to closing of stomata pore. Water, it also acts as a hydrogen donor in this process. Only 1% of total water absorbed by plant using photosynthesis. The shortage of soil water may indirectly affect the photosynthesis because the shortage of water may cause a closure of stomata. As a result, gases exchange stop. The water may cause the closure of stomata, and there is potential of the leaf become low, and the enzymatic activity may be adversely affected. Oxygen, increase in concentration of oxygen will affect the rate of photosynthesis. Oxygen acts as a competitive inhibitor for the enzyme Rubisco, and Rubisco will carry out oxygenase activity if there is a high concentration of oxygen molecule. Okay, thank you.